So it is my great pleasure to introduce Tiffany Amariuta for an in-person seminar here. So hopefully many of you have gotten the chance to connect with Tiffany already since she has recently joined us. But Tiffany did her PhD at Harvard Medical School, where she worked with Dr. Ray Chowdhury on autoimmune disease and inference of cell type specific regulatory elements. And then moved on to recently do her postdoc with Alkus Price, who actually has some connections to UCSD already as well. I think Alkus was Pavel Pevsner's PhD student, maybe? Postdoc, something like that. So she did her postdoc in the Price Lab, where she was working on statistical genetics and methods development. And she has recently joined us as a new faculty and joint between DBMI and HDSI. She's sitting over in the new building at Franklin Antonio Hall. So please stop by and say hi to Tiffany. And I will now give the microphone to Tiffany, and I'm excited to hear what she has to tell us.
Thank you so much, Melissa. All right, let me just get everything situated. All right, so hopefully everyone can see my screen who's on the Zoom and can hear me. So I'm super happy to be here in person giving this talk today. It's been quite a long time since I've given an in-person talk, so this should be fun. I want to encourage everyone to ask me questions as I go along, because I really don't want you guys to get lost in any way. And if there are questions on the Zoom, hopefully, thank you. Okay, that sounds great. So I'm really happy to be here today to share with you the work that I did in my postdoc. And then at the end, I'm going to talk about a few directions that I will be pursuing in my lab. So you can keep that in mind if you're interested in doing a rotation project with me. So today, I'm going to be talking about modeling tissue and gene co-regulation to reveal causal tissues for disease. All right. So today I really want to convince you guys that inferring causal cell types is a very important goal and that it's absolutely essential to understanding disease mechanism. So let's imagine we've performed a genome-wide association study or some sort of EQTL study where we've identified maybe a disease variant or a disease gene. That's the first step to understanding mechanism of disease action. However, if we don't know the cellular context in which this is playing out, the cellular context which actually mediates this genetic variant to causing an effect on phenotype, we really don't have the complete story. And not having the complete story is going to inhibit all sorts of different follow-up analysis, such as just if we were to do experiments, what kind of cellular context would these experiments need to be in if we're thinking about designing drugs for future interventive treatment? Which cell type should we have this drug target in the body? Especially when you're thinking of non-systemic diseases that will affect very specific tissues in the body. For example, arthritis will affect the joints. Without knowing the exact cell type and context that these mechanisms are operating in, we also don't understand the environment around that. What other genes and proteins is this mechanism interacting with? And so this is incredibly crucial. So luckily, with the intersection of genome-wide association studies, which will tell us which variants in the genome are associated with a phenotype, luckily, as I was saying, the integration of this sort of genetic association data with functional genomics data has begun to give us an insight at least to which cell types are associated with disease. So for example, you could imagine taking your top hit from a genome-wide association study, and then maybe trying to infer what sort of cellular activity is happening there. So you might have genomic tracks such as gene expression measurements or chromatin accessibility or transcription factor binding, and you can imagine having these measurements over an array of different cell types or tissues. This can start to give you some insight into what is going on there, but usually what happens is you find an immense amount of overlap between these sort of genomic tracks across different cell types. So it becomes very hard to disentangle what's truly going on there. So for most diseases and traits, and for the purpose of this presentation, I'm talking about polygenic diseases and complex traits. These are phenotypes that have hundreds to thousands of different contributions to them from different parts of the genome. So for most of these, trait-associated cell types have been identified. And in this talk, I'm going to be talking about cell types and tissues interchangeably. So for example, using gene expression data, specifically EQTL data, which means not only do we have gene expression, but we have genotype data for these individuals. The frontal cortex of the brain has been implicated as potentially the most likely causal tissue for schizophrenia, or at least in this study, it has the highest enrichment statistic based on the method that is used here. Another example is that using chromatin accessibility across different cell types has revealed that bone chondrocytes are the most associated and perhaps the most likely causal cell type for height. Using enhancer predictions or really cell type specific regulatory element predictions from my previous work, we've found that CD4 T cells, specifically the Th1 subset, are the most associated cell type for asthma, and potentially the most likely causal cell type. However, the point that I want to stress today is that despite all of these associated tissues that we've found for diseases, the likelihood that all of them are causal is actually very low. And so it's a very challenging problem if we want to think about how are we going to perform causal inference with all of this association data that we've generated. And why do we believe that there's an excess of association and not necessarily enough, or it's not straightforward to infer what is actually causal? Well, it's because there exist high correlations of all sorts of genomic measurements, meaning gene expression, chromatin accessibility, transcription factor binding, across trait-associated tissues, and therefore not necessarily all of these are causal. So if we're just looking at GTEx data, which I've shown in this slide, where we are looking at co-expression on the left. So this is just the total expression we see across individuals. We look at this pairwise heat map across different tissue types. You'll notice that the correlations are incredibly high. There's this brain region that's obviously very highly correlated with each other. But if you just look at the other tissues across the entire body, there's still an immense amount of correlation. And this is the direct result of, or this is the thing that directly produces confounding in causal inference analysis. If we look on the right, now we're talking about co-regulation. So not only are we looking at gene expression, we're looking at genetic effects on gene expression. And these are similarly highly, highly correlated across tissues. So I want to talk a little bit more about co-regulation because it's not talked about as much as, for example, co-expression is. And what does it actually do that makes it hard for us to infer something about a disease mechanism? So let's imagine we've found this disease-associated locus from a genome-wide association study. Now let's say we've tried to perform some sort of co-localization analysis. So the GWAS obviously has no cell type level information in it. It's just purely genetics. Let's say we bring in brain level genotype gene expression data. Maybe we're able to identify this gold variant that has an effect on a downstream gene, here, gene X. And let's say an oracle has told us that this has an effect on disease. So this EQTL effect that I'm talking about is specifically what happens when gene expression is proportional to the allele count of a risk allele, for example. And so here, for example, gene X's expression will be correlated, everything good? Ah, I see. I think I can just push this up probably. Ah, that looks better. Okay, great, great, perfect, perfect. All right. So this example of an EQTL I'm showing has the direct relationship where gene expression is increased proportional to how many T alleles an individual has. Now let's imagine we also take this gene expression genotype data in the heart. If the exact same EQTL exists, we would infer that this is also a likely causal tissue for this disease, because to us, there's actually no difference between these, the data from these two tissues. However, that would be the incorrect inference. It gets even more complicated when we think about linkage disequilibrium, which means that variants next to each other are rarely independent. That means they get inherited together on the same haplotypes. And so you could imagine, even if you're looking at a different cell type like muscle, and maybe it's not the exact same causal variant, the EQTL in that cell type, if it is in any linkage with an existing EQTL from a different cell type that has an effect on disease, this cell type 2 in a colocalization analysis would make you think that it has something to do with disease. So these are two different types of confounding, and I'm going to talk later today about the method we created to get around this problem. So I wanted to first provide a theoretical example of why we're interested in this sort of task. So some of you may have heard of fine mapping before. So the method we're trying to make to solve this problem is very analogous to this need for fine mapping genome-wide associated variants. So this is because when you perform a GWAS, you get disease-associated loci, but within each locus, there can be many different associated variants. And that is, again, because of this linkage disequilibrium problem. All of these variants are highly correlated in their genotype, so they all look associated. In real life, though, not every one of those variants is playing a role in disease. And so there are fine mapping algorithms that just use statistical information. They can use functional information, but the standard ones like SUSY and FineMap will just use genotyping information. The goal of these methods is to output a probability of causality, whereas the GWAS is giving you an association statistic. So what I'm trying to say here is we're trying to shift our focus from just association and enrichments to actual, what is the probability of causality in some sort of like joint model. So here, for example, we're highlighting this one variant as what's most likely to be causal, even though it was not the top hit in the original GWAS. All right, so what does this mean in tissues? So here's, this isn't, the left is real data and the right is sort of like just fake data to make a point. So our goal is to create a particular type of method that will identify the causal tissue among a group of highly co-regulated, correlated tagging tissues. So on the left, I'm showing a result from the Finucane 2018 Nature Genetics method, which is about using specifically expressed genes as a way to interrogate the associations we see in GWAS. So here, Hillary, the first author of this paper, has taken many different functional annotations that are lists of cell type specifically expressed genes in different regions of, in orange here, the central nervous system. So the association with BMI, we know that the central nervous system is involved, and this method is definitely like showing that this is a glaringly like very strong association. However, there's no way to interrogate the difference between these cell type regions of the brain. And we're really interested in creating some sort of fine mapping approach that will allow us to say, okay, it's actually the cerebellum, or it's actually some very specific cell type in which the disease mechanism is active. So I want to show a real world example of how this confounding issue is very pervasive, even in the most, even in the clearest of examples where we do know the biology of what's going on. So in this slide, I'm introducing this new term, a transcriptome-wide association study, or a TWAS. So it sounds a bit like GWAS, right? So instead of in GWAS, where we're testing the association of each of 10 million different variants, let's say across the genome or more, with phenotype, here we're testing the association of, let's say, 20,000 genes with phenotype. And the way we do that is, it's a bit tricky, and I'll get to it in a later slide, but of course, just keep in mind that we don't have gene expression measured in all of the people that we would have in our GWAS cohort. So what we do is we end up making predictive gene expression models. But again, I'll return to this later because it's a tricky concept. In any case, the idea is to find associations between a gene and a disease. Conceptually, this is described as the cisgenetic correlation of gene expression with disease. So sort of like what I showed before in the schematic, it's looking for co-localization across many variants of variants that affect gene expression and simultaneously are associated with disease. Now, we know very surely that SORT1, a gene, specifically in the liver, regulates cholesterol. When you run TWAS on the SORT1 gene across different tissues, the liver is not the tissue that has the greatest association statistic, even though we know that that is the tissue harboring the causal mechanism. So this is just an example of where causal inference of some sort would actually be very useful. And even in the the strongest of association signals that we're sure of when we are sure of the biology, there's still confounding that is occurring. So here I'm comparing on the left GWAS to TWAS on the right. And this is sort of going to be the crutch that we use as the basis of our method, because the association signals that we observe in both types of studies actually have a nice predictable pattern that we can leverage to our benefit. So in both GWAS and TWAS, the association signal, which we call a chi-squared statistic, which is approximately what you would get if you took the square of a z-score, approximately. So this association statistic has been shown to be pretty much linearly proportional to the total amount of associated or tagging or co-regulated causal effects. So in GWAS, we can plot this GWAS chi-squared, which is a value per variant, against what's called an LD score. This is just the amount of correlation a variant has with the rest of the variants in the genome. In TWAS, we can again plot this chi-squared statistic, which is one value per gene, actually one value per gene per tissue. And then we can see that there's still this linear relationship with how much co-regulation is occurring. So just like linkage disequilibrium is the correlation of a variant with all other variants nearby, co-regulation is the correlation of a gene with all other genes nearby. However, in the genetic space, not in the total expression space, but I'll get to that again in a later slide. So the overall concept is that we want to estimate some sort of slope, because with the GWAS example, we've shown that the slope is actually representative of the disease heritability. How much variation in the phenotype can be explained by genetics. In the TWAS example, what we want to do is exploit this sort of value that the slope naturally produces as a tissue-specific estimate for disease heritability. And that immediately has the implication of being a causal quantity. So we developed this method called TCSC. It stands for Tissue Co-Regulation Score Regression, to disentangle highly co-regulated tissues. And if any of you are familiar with LD score regression, this is very analogous to partitioned LD score regression. But in any case, I'm going to go through the steps. So step 0 of our method is to select a phenotype and get genome-wide association statistics for it. The next thing we do is we run a TWAS. So this is going to be performed by intersecting essentially the GWAS data and some EQTL data. And we want to have an association statistic for every gene tissue pair. So ideally, you could imagine that's 20,000 genes by however many tissues. There's about 50 tissues in GTEx. It's a sparser matrix than that because not all genes are expressed in each tissue. But yes, Abraham, question. Yeah.
I'll bet you're going to get there, but right now by tissue, we mean tissue which might be a heterogeneous group of cell types.
Exactly, exactly. It's not perfect. That's what we've got. Yeah. Yeah. And I'll circle back to that point at the end, but it's a good point because it does mess up our causal inference. So the first thing we get is this vector. This is analogous to what I was showing before with sort 1. This is sort of like the bars that was in the slide. So for every gene tissue pair, we have a chi-squared statistic. In order to perform causal inference, we need to carefully track how each of these genes is related to all of the genes nearby, and also those genes in different tissues, or technically the genetic component of those genes in different tissues. So the idea of running TCSC is to regress the values that are in light green on co-regulation scores, which I'm going to explain on the next slide, specifically how to create, across each tissue in a joint model. And so the fact that we're modeling tissues together in a joint model means we're distinguishing ourselves from association analysis, and we're finally in the space of causal inference, because now every single estimate that we have for a tissue, which will be in this dimension, will be dependent on the other. And that's very important for identifying that tissue 1, even though it's related to tissue 2, maybe that's the causal tissue, even though both of them look associated. So this is a multivariate linear regression. And then every cell in blue here is the sum of the squared correlation of the genetic component of gene expression in the gene that is represented in this row in green with every other gene expressed in that tissue. But again, I'm going to get into this in detail on this slide. So the first steps actually to estimating tissue co-regulation scores, which again are sort of analogous to LD scores, is to kind of do the steps that you would do to perform a TWAS in the first place. So like I mentioned very briefly in the beginning, we can't just simply associate total expression of a gene with a phenotype, because to find effects with a phenotype, we have these massive GWAS cohorts of thousands or hundreds of thousands of individuals, and you'll never find a study that measures gene expression in this many individuals, at least not by today's standards and expense that it takes to measure gene expression. So instead, what we have to do is create predictive models of gene expression and then apply these to the people in the GWAS cohort. So instead of total expression, we'll have a predicted value of expression. What's the first step here? The first step is to learn how genetics is associated with gene expression. So we'll go into a cohort such as GTEx. We'll look at expression across genes. And so here, let's take one gene. We will fit a model of the different like allelic counts across individuals and produce some sort of like joint model of these effects. So now we have a model. We can take that model and introduce a new set of individuals with a new set of genotypes and simply take the dot product of these to get a vector of predicted gene expression. And we refer to this as W. Now, the idea here is to compute, to estimate co-regulation, is that for every single gene tissue pair, we're doing this with reference to a particular other tissue. This other tissue can also be the same tissue because there's, and the answer is not that it would be one, because you need to take into account that that same tissue has other genes, and co-regulation can exist across genes. So what we do here is we sum up the squared correlations that we see with each sentinel gene tissue pair shown in purple on the left with every gene in a locus. And we arbitrarily define this locus as like 1 megabase, because that's what we believe will be sort of where the linkage disequilibrium falls off. And then at the end of the day, we sum up all these squared correlations, and then we have an idea of just how all of these association statistics are related to one another. If we can't model that, then there's no performing causal inference. So we implemented this as TCSC. And yes, Melissa, absolutely. Yeah, it is gene by tissue, where gene is sort of like, I don't know what the right word to say for this is, but it's not, it will be gene, every gene tissue pair concatenated together. So it'll be way more than just 20,000 genes. It'll be every single gene. Yeah, effectively. It's a large matrix. But if you take into account that not every gene will be measured in every tissue, then it's less than 20,000 times 50. Then times 50 again, because that's the number of columns. Yeah. Okay. Hope that made sense. All right. So in simulations, we use simulations to just prove that we are actually estimating the quantities that we expect and that we're able to distinguish these causal tissues from tagging tissues. So we use a previous TWAS simulator, and then we apply our method to the association statistics we get from TWAS. So on the left, I'm showing power, in the middle is calibration, and on the right is bias. So power is what we use to demonstrate how often, as a percentage of the total number of simulations, are we correctly identifying the causal tissue? Meaning, how often is this coefficient that we're measuring that I showed a couple slides ago in this multivariate regression, how often is this coefficient for the truly causal tissue significantly greater than 0? For calibration, this is assessing the type 1 error or the false positive rate. So here we, with 5% error being sort of a baseline for what we would expect for the null, we're essentially counting up the proportion of null simulations, meaning tagging tissues that are also present in this multivariate model. How often are we finding that they are not significantly greater than 0? And you might notice that, oh, well, you would expect this to be at 5%. And it's true. We would expect it to be at 5%. And it does appear that there's some conservative issue, probably with the fact that we're jackknifing, which is a standard way to approach the issue of estimating standard errors across the genome, because the genome has a lot of correlation in it. So you can't just estimate one quantity across the genome. You have to sort of remove segments and see how that changes your estimate, and then make a better estimate of the error from that. But historically, jackknife standard errors tend to be conservative, which means they tend to be larger than empirical standard errors, and that therefore we're finding conservative type 1 error, which is okay because it doesn't seem to really affect our power. All right, let me just move. Well, I can't move the panel that's on my screen. You can't see it, so it's okay. So on the right-hand side, I'm showing bias. So this is how well are we estimating this quantity of tissue-specific heritability? How well are we estimating this coefficient in the multivariate regression? So in these simulations, we've set this value to 5%. And you can see as we're increasing the EQTL sample size, which is effectively increasing the quality of the gene expression data, the quality actually of which we can build tissue co-regulation scores, the quality with which we can distinguish tissues from one another. It gets better and better as we're increasing this sample size, which is expected. And there's, of course, some bias if you don't have enough individuals. And that's what's shown in purple. And actually what's shown in gray, which is really hard to see, is the bias on the tissues that are not causal. So the nice thing is that the average value for this heritability term is not significantly different than 0 for most of these estimates, which means we're succeeding in terms of the simulations we're running. Yes, Melissa. Yeah. Yes, perfect. So the setting for simulations was 10 tissues. And the idea was to create correlation or co-regulation among them that is directly representative of GTEx. So it was completely modeled on what levels of co-regulation we would see between tissues at different sample sizes. And so each sample size is actually representative rather than just like the truth being representative, because it's hard to estimate the truth with GTEx. You could take the largest tissue potentially and like do that, but we decided to do it on like an estimated per sample size basis. And the last thing I wanted to say is we also have modules of tissues. This is in the additional simulations that I kind of mentioned at the bottom of the slide, where it's effectively representing brain as like a big component of GTEx tissues versus just like stomach versus muscle, which maybe are like less related than brain. And we show that the model works regardless of how much co-regulation you have. All right, so now that our simulations worked really well, we applied this method to real gene expression and trait data, meaning like real EQTL data and real genome-wide association studies. So to do this, we built these gene expression prediction models, which is again, like I explained, the first step to doing a TWAS across 48 different GTEx tissues, where we're only retaining heritable protein coding genes. So these are essentially the only genes for which we could build a good genetic predictive model. Otherwise, what's the point of you're associating like a null model with a trait and shouldn't find anything. And if you do, it doesn't mean anything. The second thing is we chose 78 independent European GWAS summary statistics with large average sample size. Most of these came from UK Biobank, but some of them were just from like publicly available summary statistics. And then as a result, we applied TCSC and we find 27 causal tissue trait pairs. So this is the heritability analysis that I've been talking about for this entire time. In the second bullet point, we found 30 causal tissue trait covariance pairs. So I didn't talk about covariance in this presentation due to time, but basically there's a way to adapt this model that we've created, which uses TWAS chi-squared statistics to, instead of interrogating heritability and partitioning heritability across tissue, you can partition the covariance or genetic correlation of two traits across tissues by replacing these chi-square statistics with a z-score multiplied by another z-score, but from two different traits. So we showed that that works too. And that's pretty cool because the application of that is you can imagine genome-wide, you'll have two traits that have no observable genetic correlation, but maybe it's just because it's cell type specific and it's washed out genome-wide. So there are some instances where we do find this, and I find that those are the most compelling applications. We're also able to increase the specificity of known tissue trait associations, and I'll get to that later. So for example, instead of just identifying that central nervous system is involved in BMI, we can say the specific region is most likely. All right, so here's an overview of these 27 tissue trait pairs that we found at 10% FDR. The value I'm plotting here is this pi t prime statistic that we made up for what TCSC outputs. The value is directly this coefficient that I talked about estimating in the regression, but scaled based on the heritability of the trait, just so that all the results across different traits could be more or less compared to one another. And then I'm highlighting 3 examples that are the more novel, I would say, than the others. So, on top of re-identifying that, yes, of course, whole blood is probably the most likely causal tissue for a bunch of blood traits or immune diseases such as hypothyroidism, or that liver is the causal tissue for all of these lipid traits. Some more interesting results were that we found glaucoma. The causal tissue, most likely causal tissue for glaucoma seems to be the aorta artery. Now, keep in mind, there's no eye tissue in GTEx, so this could very much be an issue of tagging for the sake, in the sense of the tissue is absent in the data set, and so my method is going to identify the best proxy. Regardless, though, it's interesting because high blood pressure is a very known, recognized risk factor for glaucoma. So there might be something actually biological there. We also found that FEV1 over FVC, which is the forced expiratory volume, so it's how much air you can push out of your lungs divided by your capacity of your lungs for air. We found that the esophagus muscularis is potentially the causal tissue there. And that kind of makes sense because it might govern the strength of the muscles to expel that air. And the interesting thing is we also had GWAS for just the numerator and just the denominator, and we were able to rule out that there's no association or causal link between esophagus and FVC. So it does probably have to do with this more active phenotype versus the passive lung capacity phenotype. Lastly, we found that the heart, specifically the left ventricle, is potentially causal for platelet count. And this is interesting because we know very well platelets cause blood clots because they're recruited when there's damage done to blood vessels. And the left ventricle is responsible for pumping blood out of the heart. So you might think that, when you're collecting these individuals for GWAS and you're measuring their platelet counts, this is done in serum and whole blood, for example. And therefore, the heart is probably playing some role in at least modulating these levels. All right. So like I said, we also increased specificity of known tissue trait pairs. So for example, previously, the field had some idea that adipose was involved in HDL. But specifically, we found that this causal link only exists for subcutaneous adipose versus this visceral omentum subset of adipose tissue. And with further reading and a literature search, because we weren't unfortunately able to validate any of this beyond statistical analysis, it seems that adiponectin may be at the center of this because it is specifically associated with subcutaneous tissue and has nothing to do with visceral. Very similar thing we found for waist-hip ratio, which is adjusted for BMI, whereas you might expect to find associations with other metabolic tissues. And then lastly, this is the example I've been talking about since the beginning is BMI and the central nervous system are different brain tissues. So we were really able to pinpoint that brain cerebellum seems to be the most likely causal tissue among GTEx tissues. But then again, they're like Abraham brought up before, these results are highly dependent on the cell type composition of any of these tissues. So you can imagine that each one of these tissues is made up of different cell types, and depending upon the concentration of maybe the most likely causal cell type, if it's higher in one tissue and not the other, TCSC might pick up on that tissue, even though the tissue is wrong, but maybe the cell type is more represented. So that's another very difficult thing, and is very data dependent. Yeah, Melissa. Oh yeah, we downsample. Yeah. Oh yeah. So I'll repeat the question. So Melissa asked, how do we deal with different GTEx tissue sample sizes? We downsample to make them all the same, or we meta-analyzed brain tissues, for example, to achieve an appropriate sample size. However, asterisk on that, It's a, yeah, the asterisk is that, oh, what do I need to move something? No, that's a huge issue. Oh, yeah. Yeah. We're downsampling everything to the same exactly 320 individuals is what we considered for every tissue. The meta-analysis asterisk that I'm mentioning is that in brain, in all of GTEx, you have only 100 and I believe it's 80 unique individuals. So it's hard to say that this meta-analysis is completely independent, but we did meta-analyze them as if they were separate samples. However, the genotypes are redundant. So it's a bit of a gray area, but we try to achieve this fixed sample size, which is why brain cerebellum has a little dagger next to it, because it is a composite tissue of the of two different types of cerebellar tissue that's represented in GTEx. Yes.
I don't think you're actually going to tell us that they again bipolar or caused by blood. But so why that blood cell?
Yeah, it's interesting. So specifically for like major depressive disorder, the top TWAS hit is actually like some sort of cytokine gene, which obviously implicates the immune system in blood. And I'm thinking maybe that signal was just very, very strong, and for some reason, the whatever signal we do have in brain is somehow washed out. But later I will show a brain specific analysis where, it basically allows us to interrogate for brain related phenotypes. Let's just remove the chance that like any of these other spurious correlations could potentially throw off our results, and then see what is the most likely brain tissue. Yes, Terry.
So in on the previous slide, glaucoma.
Uh-huh.
So primary epidemic glaucoma, this factor is high blood pressure, but none of this factor is low blood pressure.
Yes, I just learned about that.
So what's going on for you?
Yeah. That's a great question. So I'll just repeat it really quickly. So Terry mentioned that not only is high blood pressure a risk factor for glaucoma, but so too is low blood pressure. And yes, I was just talking with some folks over at Shiley a couple of days ago, and I was talking to them about this result, and I heard the exact same thing. And it's unclear what to make of this. Yeah.
Minimizing progression.
Okay. Oh, that's interesting. So are you saying that like progression severity of glaucoma is not affected by blood pressure, but maybe the binary existence of the phenotype could still be? Or yeah, it's interesting. In fact, some folks over at the Broad, when I was presenting this work back in Boston, they were they wanted to share with me some like histology images that they had that could maybe help me figure out if it is this thing about severity or if it's more of like a binary thing, but haven't followed up on that. But it's interesting. It can also, again, there's no eye tissue in GTEx. This could just be a proxy causal tissue. So it's hard to say. Yeah, but of course, if you think of the optical nerve and being affected by blood pressure and then those vessels, then you could, yeah. Okay. Sure. All right, great. All right, so moving on, we benchmarked our method against other strategies. The one on the left it actually claims to perform some sort of causal inference, whereas the one in the middle is still strictly an enrichment or association strategy. But the take-home message here is that both of these methods produce lots of correlations or lots of associations across different tissue types. And the tissue types that I'm showing in this slide, the ones that are represented in pairs that are separated by solid black lines are ones that are highly co-regulated. So if we look at the TCSC results, for every pair, the left tissue is the one that I've identified in my analysis, and the tissue on the right is the one with the highest amount of co-regulation based on the EQTL effect sizes in GTEx. And so you can see just based on the other methods that they really don't have any ability to distinguish between these highly co-regulated tissues. This RTC colloc method is a way of co-localizing EQTL data with GWAS, but normalizing it for tissue specificity or the lack of tissue specificity. They call it tissue sharing. So the more your co-localization signal is shared across tissues, the less it contributes to the model of this enrichment statistic. However, you still see for, let's say, diastolic blood pressure, many tissues across the body are associated. All right, so moving on to this brain-specific analysis that I hinted at earlier. This is a really interesting case to see if TCSC still works, because brain tissues are more highly co-regulated with one another than random tissues across the body. And of course, we're comparing this to LDSC-SEG, which, like I said before, is not a causal inference method, and is still either not able to identify associations with any tissue, or is not able to distinguish closely related brain tissues. So actually, I noticed that the result for MDD is not shown here, and maybe that's because we didn't detect anything significant. So I'll get back to you on that, Abraham. Yeah. But yeah, so the idea is that essentially we are able to distinguish really highly co-regulated tissues, such as the brain cerebellum from the cerebellar hemisphere, or the frontal cortex from just plain cortex, as is labeled in GTEx. And the follow-up to this is that you might be worried that, okay, maybe there's some randomness in TCSC that's causing it to select one tissue over another, and maybe it's not actually true, the one that you're selecting. Maybe there's some regularization, or lasso, for example, which will randomly choose one of two correlated features. But actually what we show is that in simulations, if we model two identical tissues that have the exact same EQTL structure and add just a little bit of noise to them to make sure there's no collinearity in the model, we're actually getting very equal amounts of heritability estimated for both of these duplicate tissues, which kind of reassures us that in the last slide, it's not just random that TCSC is picking one closely related brain type over another. Yeah.
Related question.
Yeah.
Question, and so what you're talking about. I guess my sense and the detect brain data is that even if you adjust for sample size, like some tissues are just cleaner.
Yes.
than
Yeah.
others,
Absolutely.
Therefore, will be like better powered. And I think that could cause you to just choose the tissue where you have better power.
Yeah. Okay. So I'll just repeat the question for those on Zoom. So Melissa asked if even if you were to downsample like we did the tissues so that they all have the same sample size, if the tissues have different quality, and quality can mean something like maybe they're just one's more homogeneous than another, maybe one is more difficult to collect, or any number of features can lead to worse gene expression data. Yeah, absolutely. That can cause issues with the inference. And I'm thinking of like the covariates that we do have available to us in GTEx, if they can even be useful in this manner. I'm not really sure. I mean, at the end of the day, what do we have? We have obviously like genotyping PCs, which is just from the genetics, but we also have these peer factors, which is from the gene expression data. And honestly, now I'm just thinking about it, it could be that when there's more variation in the gene expression for good or bad reasons, right? Biological or non-biological, maybe there's a correlation with like how many peer factors explain a certain amount of variance or something like that. We haven't looked into that, and we just assume that the quality is the same, but that's definitely something that can contribute to issues. Yeah, Alon.
Using single cell RNA cells
Mhm.
to identify the structure of the tissues. Is it something you consider using in order to identify the different components within the tissue?
Yes. I think this is my next slide. Yeah. So we've really, yeah. We've really applied TCSC in this proof of concept, like applying it to GTEx tissues, which are the most different type biological samples that you could possibly get, right? They're from all different parts of the body. And good, it works there. But it'd be so interesting if we took it into more complicated or finely resolute systems where we can make more meaningful inferences. Like, for example, it's really not interesting just to identify that diastolic blood pressure is probably associated with the aorta. Like, okay, we all know that. But in the future, I'm really hoping to apply TCSC to cell type level data. So here I'm just showing like hematopoietic lineages. It would be super to disentangle these, because for autoimmune diseases, it's very unclear what the contributions from each is, especially in heritability units. That's something that hasn't been done before, but obviously there's loads of association studies out there using functional data. Some of my previous work included. So it would be super to apply it on that front. And then even on the single cell level would be really great. Some statistical challenges will be that the fact that we model EQTLs differently when we're looking at single cell data, and that's okay. Or you could take the approach of pseudo bulking, and then you can use standard pipelines. But yeah, ideally, we would love to perform causal inference at all of these levels, because that will really make more precise mechanistic inference. And also, TCSC is a genome-wide method. It would be really cool to develop a locus-specific method, because not every gene for every disease is going to act through the same cell type. And so we would really want to say, like, what is the composite contribution from all these different cell types, and where is it in the genome? All right, so to conclude on this, one, we found that, or the field has recognized that co-regulation of genetic effects across tissues prevents the direct inference of causal tissues from colocalization or association analysis. Second, we've found that indeed co-regulation scores and TUAS statistics can be used together to infer the causal tissues mediating disease. And TCSC seems to identify potentially new, maybe biologically plausible tissue trait pairs. But again, we are, at the end of the day, this is just a statistical method. And of course, we require immense amounts of biological follow-up to make real statements about what is mediating biology. But we think this is a good first step, at least, away from these association analyses where you really can't do more than what the statistics tell you, and you can't infer beyond what the largest hits are or the greatest enrichments are. So in terms of future directions, we've touched on some of these, but one, I want to mention that our results might seem conservative. We looked at 50 tissues, almost 80 complex traits. We still only found 27 causal tissue trait pairs. Seems like a pretty low number given the total amount of combinations. However, since we're looking at GTEx data, there are some limitations, such as the fact that we're looking at steady state gene expression rather than, let's say, stimulated immune cell types, which are going to be the relevant cell types for immune-mediated diseases. We're also looking at tissue level data, which has all these issues with heterogeneity of cell type populations that are within it. And we're also looking at healthy individuals. And it's true that EQTLs change between when you're looking at cases versus controls, at least in some cases. And this can give us an entirely different impression of how the genetic association signal for disease relates to gene expression. Second, again, this is reiterating, so as cell type proportions within tissues can affect our results, we hope to apply TCSC to finer resolution cell types so that it's not just this latent variable of like what cell type is most represented within the tissue that seems to be making that tissue just perform a little bit better in the model. And lastly, to better infer causal tissues in polygenic diseases, as you might have noticed that most of the associations or causal links that I showed were for complex traits. We didn't get a lot of results for polygenic disease, and that was a bit unfortunate. And I was hoping for more, given that we have a lot of autoimmune diseases represented. But of course, even the result of whole blood wouldn't have been interesting, but it still wasn't able really to pick up that signal. So that's something very interesting, and potentially the signal is washed out because, again, you're looking at steady state whole blood, lots of different immune cell populations, rather than lymphocytes, which is only going to be 1 or 2% of whole blood. So ideally, we'd like to apply this to case control EQTL data at the proper cellular resolution. All right, so I just want to wrap up by giving you guys an idea of the different research directions I'm interested in pursuing in the lab in case anyone would like to, for example, do a rotation project with me. So the focus of my lab is by and large the statistical methods development for population level genetic inference. So I'm really interested in taking genetic data across multiple populations and creating finally joint models rather than just stratifying individuals based on their genotypes and then running like all these downstream analyses. So one example of this is the development of multi-population genetic risk scores, specifically for gene expression, because I still am interested in this sort of like a TWAS framework where we're looking for disease associations based on how the effects are mediated through gene expression. So for example, here, an example of a model would be to create a population-specific PRS that, due to sample size restrictions alone, would not perform well, but could be modeled as a function of genetic data across individuals across the entire globe. Compared to what we currently do, which is, again, stratifying individuals. Second, so top right, I'm really interested in somehow getting trans variants or trans EQTLs into these gene expression prediction models. We know that cisgenetic variation is, of course, by and large, what people look at when they look at genetic effects on gene expression. However, the contribution from variants across the entire genome contributes twice as much variance as this cisgenetic variation. Of course, the statistical burden is very high to identify any one individual effect, but because we're talking about gene expression in these predictive models, in this predictive model space, we don't need the statistical confidence to identify any one effect. We kind of just need the batch effect to be meaningful. And so I'm hoping to leverage gene co-expression information, protein-protein interaction networks, all sorts of things that will point us to other genes that the sentinel gene might cooperate with to select their cis variants as candidates for trans EQTL. On the lower left, I'm interested, again, this is sort of what I discussed about, so I'll just say one sentence. This is about applying TCSC essentially at the cell type level. And this can have statistical challenges because there's more co-regulation at the cell type level. And especially if we're looking at single cells, then the whole way of looking at EQTLs is different. And so this will require a lot of methodological development. On the lower right, finally, I'm also interested in, so the theme here is sort of like getting extra variation into these gene expression models. So the last theme is getting environmental variance into these gene expression models. And recently there have been at least one or a few biobanks that have more carefully cataloged environmental exposures than before, because traditionally it's been very hard to look at environmental factors because of the way that they've been measured or the fact that they are from survey data, and there's just a lot of noise, and honestly results have failed to reproduce well in other studies. And so here's an example of where it's not only your genetics that will give you a predisposition to a certain gene expression profile, but this genetic predisposition in combination with an environmental effect will have a particular increased risk, let's say. And anyway, this can be really helpful down the line in terms of preventive medicine or even interventive medicine. If you want to say, all right, given someone's genetic predisposition, what can we recommend they not do in their life? What exposures would be particularly detrimental to people with a specific PRS or genetic profile? So I'm thinking about these areas as I transition into this independent position, which I only started a couple months ago. So I just want to thank my former advisors and some lab mates, my labs over at Franklin Antonio Hall. So feel free to just drop by and say hi. We're on the second floor. Thanks. Yeah, I'm happy.
From the internet, and Timothy Auger is asking, and maybe can you read it after I say, have you run TCSC with STEM or IPS cells?
I have not run TCSC with STEM or iPSCs. That would be honestly a great direction to go. And we've only applied this to tissue level data. And again, because of all the limitations of tissue level data, our inferences are severely limited. But at this point, it's just been a proof of concept. So yes. Yeah. Yeah. Yeah, thank you for bringing that up. Sometimes I have time to talk about reverse causality, so I'm really happy you asked that question. So in theory, since all of the entire model is based on genetics, even if the phenotype or the effect of the gene expression would change some other intermediate phenotype, it's not going to change the genetic association. So only if there was some sort of like shared genetic effect on both of these phenotypes, then there's really nothing we can do about it. But if there's separate genetic effects, then there shouldn't be any residual reverse causality left over. Yeah. Since again, we're talking about the genetic component of gene expression rather than total gene expression. If we weren't, then absolutely that would be a problem. Yes, in the front.
was wondering, does it mean that it's going to definitely work in a monogenic or a chromosomal repeat, extra chromosomal phenotype?
So are you asking about like you said the development of just methods in general for polygenic traits, or?
Monogenic, if you want to validate.
Mhm.
Like extra chromosomal, like Down syndrome.
Sure, sure. So that's an interesting problem because the reason why this method works is because it's taking these genome-wide averages of signal. So it's not
Even
necessarily straightforward.
they have different expression, profile.
Yeah, definitely, definitely. I'm just thinking that right, you at least need different observations across a dimension. So that dimension could be tissues, even if it's not loci. So I think in theory, yeah, it should work. That's a great idea. Thank you. Oh, yes, in the back.
What if you are like
Yeah, this is a great question. So basically considering variation that's not germline, right? So this is these are population level models. So we're only looking at common variants, meaning they have at least 5% allele frequency in the population. That this sort of thing, looking at non-somatic variation, introduces other statistical challenges and power issues based on, a lot of them could be singletons, for example. So it's not necessarily straightforward because again, we're taking these associations over like hundreds of thousands of individuals, and that really assists with our power. So I would have to say, I'm not quite sure or optimistic that that would work, but it's something to think about, and yeah, potentially in the future we'll address that. Yes, Terry.
I'm going to ask you a computational question, but it's motivated by glaucoma. So the SNP that is the most associated with primary open-angle glaucoma,
Mhm.
one that's been studied, is the same SNP as has been found as protective against glioblastoma, and it's also risk for cardiovascular disease.
Mhm.
So where this goes is you've got the same exact gene, CDKN2AS1 and then CDKN2E, which is nestled in the first intron of CDKN2AS1. So basically what you have is the same region being implicated in multiple diseases through multiple GWAS. That's really what's going on here. Now, if you take your aorta finding that light, then it starts to make some sense. So I'm wondering if you could gain tissue power, not just one GWAS to TWAS, but actually look at the GWASs that are implicated in the same areas of the genome. Or, for example, type 1 diabetes and rheumatoid arthritis would be another.
Sure. Thanks, Terry. Okay, so I'm just going to try to briefly recap the question. So the idea is we're considering a single phenotype, at least in the results that I showed, and gene expression. If maybe we can increase the biological plausibility of our results having integrated multiple GWAS. We've done this for the 2 GWAS case, where we are partitioning covariance or genetic correlation across tissues, but you could imagine this in like a multi GWAS sort of structure, because again, at the end of the day, you're just modeling how these association statistics are correlated with one another, and it should generalize to more than one phenotype, I would think. It wouldn't exactly be correlation or covariance, but that's a great idea because then you could pinpoint where the signal is coming from. Yeah, that would be great. All right. Thank you, everyone.